Okay, so we talked about gated transport that was going to allow bidirectional transport in and out of the nucleus, and it could take both folded or unfolded proteins. Now we're going to talk about translocation. We're going to start by looking at translocation in the mitochondria and the chloroplasts. So remember that these are the organelles that evolved through the process of endosymbiosis. They are not part of the endomembrane system and they have a full double membrane. So this means that if we're going to try to put something in there, we're going to move a protein in there, we have to get through both of the membranes. So the process of importing, it is a one-way transport, importing proteins into the mitochondria and chloroplasts is going to start with an N-terminal signal sequence. So this is, remember, the very first thing made in a protein is the N-terminal, okay? So immediately upon translation, we begin by making the signal that says, take me to the mitochondria. So this is a signal that is going to direct this protein in an unfolded state to the mitochondria. In the outer mitochondrial membrane out here is going to be a receptor. The receptor protein can bind the signal sequence. So remember that we said earlier, these sequences can't really do anything if there's not a receptor to recognize and bind to them. So the receptor lies in the outer mitochondrial membrane. When it binds to the signal sequence, the receptor, the protein, and this channel that is attached to them, this yellow channel here, this whole thing is what's called a TOM, which means transmembrane complex outer membrane. So TOM. When TOM binds this protein, it begins to laterally diffuse through this outer membrane. So remember that this is a phospholipid bilayer and we have lateral diffusion. It's going to diffuse, sort of dragging this protein with it along the outer membrane until it comes in contact with what's called a TIM, which is shown in orange here. TIM means transmembrane complex inner membrane. Okay. Now TIM also has a channel in it. When the TOM and the TIM come together, these two channels actually line up and the protein is going to be able to move into the mitochondrial matrix. It can't move until the TOM complex comes in contact with the TIM complex. Because if it were to move through this right away, it would end up here in the intermembrane space, and that's not where we want the protein to be. So we need to bypass both of the membranes at the same time. In order to do that, we have a complex in the outer membrane, a complex in the inner membrane. They come together, and that allows the protein to move through into the matrix. Now, once it moves into the matrix, you're going to have the overall folding take place, and it will usually lose that import signal sequence. So we'll have a signal peptidase that would come and cleave that off. And now we have a mature mitochondrial protein here. There are several chaperones that are not shown here for sort of simplification, but there are chaperones that actually help keep the protein unfolded while it's sort of snaking through the two complexes. Chaperones that help force it in this direction instead of moving backwards. So there's definitely some other proteins that are in here, kind of assisting along the way. But the major components are the TIM complex and the TOM complex that need to come together. Only the TOM complex has the receptor, obviously, because the protein is out in the cytosol, so it wouldn't make any sense to have a receptor on the inner complex, only on the outer complex. Protein translocation is also used to move proteins into peroxisomes. So remember that peroxisomes are going to be specialized vesicles that deal with the buildup of hydrogen peroxide, which is toxic and needs to be detoxified in order to continue on with the cellular processes. So you are actually going to work on peroxisome transport for your assignment this week. We use protein location into the ER as well. This is a much more complex process than the mitochondria, chloroplasts, or the peroxisomes. So this is what we will be starting with in our next module.